Hello everyone, so in the last class we talked about the living world, in which we discussed about the different characteristic features of a living organisms like growth, metabolism, reproduction, consciousness, cellular organization, etc. Then we talked about binomial nomenclature, in which we talked about the naming of different organisms with respect to their genus and their species. Then we discussed the taxonomic hierarchies, in which we talked about different groups of taxons like kingdom, phylum, class, order, family, genus, species. Then we discussed about different taxonomical aids like herbarium sheets, like geological parks, like keys, like botanical gardens, etc. Now in the next chapter, that is biological classification, we discussed the different classification systems and different types of classification systems like natural system, artificial system and phylogenetic system. And then we also talked about different types of kingdoms like 2 kingdom classification, 3 kingdom classification, 4 kingdom classification, and finally the 5 kingdom classification, which was given by R.H. Whittaker. Now in this part we'll discuss individually. Like we'll start with the first part, which is Kingdom Monera, then Kingdom Protista, then Kingdom Fungi, Plantae and then Animalia. So this will be our sequence. So I will start the first part, which is Kingdom Monera. You have to make notes along with me. Okay. So Kingdom Monera. Please give a heading, Kingdom Monera. Okay. Now Kingdom Monera comprises of organisms which are called bacteria. You have to make notes. Please note this. Okay. You have to write along with me. So, they comprise of bacteria. Bacteria are the only members of Kingdom Monera. Now, bacteria are characterized by their unicellular nature, and they are prokaryotic. They are unicellular and prokaryotic. Now, unicellular means they have single cell, okay, and prokaryotic means that cell has a nucleus which is ill developed or is of primitive type. Okay. So bacteria looks like this. If I draw a bacteria, you can see that there is a nucleus. Actually this nucleus is specifically called nucleoid. Okay, nucleoid. Why it's called nucleoid? Because oid means just like. It is not exactly the nucleus, it is like the nucleus. It has not got all the functions which are present in a nucleus. It has got some functions, and hence it is just like the nucleus. It is not a perfect nucleus. Okay, so if I say nucleoid, I mean there is a structure which is similar to nucleus. It is not exactly the nucleus. So nucleoids are present in case of prokaryotic organisms. And bacteria is one such example of prokaryotic organism. Okay. Apart from this nucleus, we have some other structures as well. Like here we have some. If I draw this like this, suppose if I draw it like this, this is the nucleus, and outside the nucleus there is a structure which looks like this, and this is known as the plasmid. This is known as the plasmid. Now, what is plasmid? We will discuss the plasmid later. At the moment, you should write it is the extranuclear DNA. Extranuclear DNA. Extranuclear means it is present outside the nucleus. The DNA which is present outside the nucleus is called extranuclear DNA. Okay. And then there is a cytoplasm. And then this cytoplasm can have some structures called ribosomes. Okay, but it is not required at the moment. So just write these points like plasmid, cytoplasm, nucleus, etc. Okay, please copy this part. Pause this video and copy this part. Okay, I'm erasing this part. Okay. The next thing is the classification of bacteria. So we can classify bacteria in a number of ways, but I will classify them into 3 categories. Or I can classify bacteria with the help of 3 categories. So I will write classification of bacteria. This is classification of Kingdom Monera बोल सकते हैं, or you can simply write classification of bacteria. Now classification of bacteria can be done in three ways. The first way is based on shape. It is based on the shape of the bacteria. The second way is based on the presence or absence of flagella, which is called flagellation. And the third way is based on staining. Please note that apart from these three, we have some more categories as well. We can classify bacteria into further categories, but for the time being, I will concentrate on these three terms: shape, flagellation and staining. Okay, now I will start with the first one, shape. Okay, now based on shape, bacteria can be of four main types. Okay, four main types are: the first one is called coccus. Coccus is spherical in shape. Okay, the next one is called Bacillus. The next one is called Bacillus. It is rod shape. Okay, so I will write spherical here, spherical shape. This is rod shape. The next one is called Vibrio. It is comma shaped. So I will draw the shape here. It is comma shaped. You can write it comma shape. And the last one is called spiral. It is spiral in shape. Okay, spiral, like this. You can have a number of examples of coccus like Staphylococcus, Streptococcus, rod shape, Bacillus group of bacteria, then Vibrio cholerae, which is the causative agent of cholera, then spiral, spirulina, which is used by the space travelers. Okay, space travelers eat this spirulina and it is rich in protein, so it is used as food for space travelers. Okay, you can note these points as well. Okay. Coccus, Staphylococcus, Streptococcus, they are pathogenic bacteria. Bacillus, rod-shaped bacteria. So example is Bacillus group of bacteria, Vibrio group like Vibrio cholerae, which causes cholera disease. Okay, spirulina, which is used by the space travelers as food capsules. Okay, because they are rich in proteins. That's it. This is the classification based on the shape. Please note this. I will erase this part. Okay, I hope you have written this. I am erasing this. Okay, the next part is about the flagellation. Now, flagellation is very simple. If this is a bacteria, suppose, and if it lacks flagella, that means there is no flagella present in this particular bacteria. In that case, let's draw it like. Suppose this is the first one, this is the second one, in which there is single flagella. In this, there was no flagella. Okay, the third one has a tuft of flagella attached at one end. Okay, and then the fourth one has a tuft of flagella attached at both the ends. Okay, and the last one has flagella attached throughout the body. So I don't have space here. I will draw it. Okay, so it has flagella all over its body. So let's name them. This bacteria which lacks flagella, this is called A. trichus or A. trichus. The next one which has just a single flagella at one end, this is called monotrichus or monotrichus. Mono means single. A means without. Okay? Trichus means flagella. Now, this condition, this is called LOFO. This is called LOPHOTRICHUS. This is called LOPHOTRICHUS. And this one is called AMPHITRICHUS. Amphi means on both the sides. On both the sides, AMPHITRICHUS. Okay? This one is called, PERITRICHUS. PERITRICHUS. So it has flagella all over its body parts. Okay? This is called peri, throughout the body part. That's it. This is the classification system. You just have to remember the names: Atrichus, Monotrichus, Lophiotrichus, Amphitrichus, and Peritrichus. Please copy this part. Okay, I'm erasing this part. Alright. So the next part is about the staining process. So I will erase the complete whiteboard because I need some space. And please give the heading staining. Staining. Stain literally means giving some colors. Okay, staining. So you are giving some colors to the bacteria, because bacteria are colorless. They need some colors. You need to put some colors in their lives. But for that you have to take lives of bacteria. You try to put colors in their lives, but in the process you are taking lives of bacteria. That means bacteria will die if you put colors in their body. Okay. Anyways, this is not always true, but in most of the cases this is true, that bacteria will die after staining. In most of the cases. Okay. Now if you classify bacteria on the basis of staining, you will get two categories. The two categories are, the first category is gram positive. The second category is gram negative. Positive and negative are two different terms. Positive means you are getting some results. You are attempting something and you are getting some results. That is called positive. And if you are not getting results, that is called negative. So that's the basis of gram staining. So here you get gram positive and gram negative, two different categories. Let's see what is the basis of this classification. This is very important, this is based on the cell wall composition. This is based on the cell wall composition of bacteria. This is based on the cell wall composition of bacteria. Now what happens in some bacteria, let's say this is a bacteria and it has got a single layer. Okay, this is single layered bacteria. What is this layer made up of? This is made up of peptidoglycan layer. Okay, and what is this actually? We are talking about the cell wall, single layered cell wall. The cell wall of this bacteria is made up of peptidoglycan or peptidoglycan. But there are some groups in which there is an additional layer apart from this peptidoglycan. So if I draw this second group, let's say this is the bacteria, this is its cell wall, which is double layered or bilayered. So I will draw an additional group here. So you can see, if this is the cell, let's say this is the cell. Okay, again, if this is the cell and cell is covered by two different layers. In this case, the cell was covered by a single layer, but in this case, you can see the cell is covered by two different layers. Two layers, okay? I can write two layered cell wall. Okay, now these two layers are made up of two different components. The first component is obviously the peptidoglycan. The first component, that means this one. This is made up of peptidoglycan, which is the inner layer. The inner layer is made up of peptidoglycan, while the outer layer is made up of LPS. Now, Let's expand this term. LPS. LPS means lipopolysaccharide. 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 Now, lipo means lipids and polysaccharide means sugars which have a number of units. Poly means a number of, saccharide means sugars. So this is a mixture which is made up of two different biomolecules, lipids and sugars, polysaccharides. Okay, so these two layers protect this cell in case of this group of bacteria. While in this group of bacteria there is a single layer. So that's the difference between two different types of bacteria. Now I don't know whether this one is gram positive or this one is gram positive. I don't know this. So for that I need a process which is called staining. Okay, which is called staining. So let's start the process. Please copy this part first and write one more point. Please add this point in a note. Make a square and write note. Make a square and write note. LPS is soluble in ethanol. I hope you know about ethanol, C2H5OH. It is in chemistry. Class 10 mein padhe hoge. Ethanol. Ethanol is a kind of alcohol which is used in biological labs and it is used as a sterilizing agent. Okay, it kills bacteria normally, but at the moment we don't need to kill bacteria. Actually, this LPS is soluble in ethanol. Okay, that's the point you need to remember. Please copy this part first. I will erase this. Please pause the video and copy this part. Okay, done? Okay. Now we'll talk about the exact process by which we can determine whether a bacteria is gram positive or gram negative. I will need two stains. So there are two different types of stains. Stains. Stain literally means colors. Okay, so colors are of two different types. The first color which is used in the process is called the primary stain, and the second color is called secondary stain. Primary stain and secondary stain. Which is used first is called primary, which is used later, that's called secondary. First and second. That's it. Now, in our process, which was called gram staining. Now, it's called gram staining because it was discovered by Christopher or Chris Graham. Yeah, sorry, not Christopher, it is Christian Graham. Sorry, Christian Graham. You just need this term Graham, Christian Graham or C. Graham. Okay. So he was a scientist, and on his surname you have this process, gram staining. Now, primary stain, which is used here, is called crystal violet. It is called crystal violet, and the secondary stain is called safranine. Safranine. Please remember this, that crystal violet is deep blue or violet in color in appearance. Okay, it is deep blue or violet in appearance. On the other hand, safranine is red to pink, reddish pink, or I can say red or pink in appearance. It is based on their concentration. Okay, so remember these colors. Now what happens here is, let's see, this is a test tube. Okay, this is a test tube, and in this test tube you have some bacteria. You have cultured some bacteria. So it's a solid mass of bacteria or bacterial culture. Bacterial culture means you have cultured some bacteria. Not culture civilization kind of bacteria, no, not like that. Culture means you have cultured some bacteria, you have brought some bacteria from some distant location and you are growing it in your laboratory. Okay, so that is solid mass of bacterial culture. You will know about this when you go in class 12th. There is a chapter on biotechnology. We will discuss this later. So for the time being, just remember this, that this is a test tube, and inside test tube you have some bacterial culture in which millions of bacteria are there. Okay, now you take a loop. This is a loop. loop looks like this pen, and there's a structure, there's a spherical structure like this. You'll take this loop inside the test tube, and just like this, you'll take the test tube, you'll take a loop inside a test tube, and you'll pull out some bacteria. So when you take out this loop from the test tube, you actually get some bacteria which actually get trapped in this loop. Now some bacteria are trapped here. Okay, now place these bacteria on a slide. Okay, just rub it or make a smear it here. This is called smearing. This step is called smearing. Okay, this is a slide, glass slide. You have seen a glass slide, I think. I hope you have seen. Okay, so this is a smear. Okay, and this smear consists of a number of bacteria. Now, this smear has both kind of bacteria. It has gram-positive as well as gram-negative bacteria. Okay. So now next step is to apply primary stain. You have to add some primary stain into it. Now the primary stain in our case was crystal violet. I will write CV for crystal violet. I will use the short term CV. Okay. Primary stain crystal violet. So when I add this over this bacterial culture, okay, what will happen? The bacteria will take up, all the bacteria, gram-positive and gram-negative, all the bacteria, they will take up this crystal violet, okay, and they will become colored. Okay, if someone throws colors on your body, then you will be colored. That's the case in this bacteria. So in this case, what happens? All the bacteria becomes colored.